Right now, without you thinking about it, your kidneys are filtering your entire blood plasma about 60 times a day. They pull roughly 180 liters of fluid out of your bloodstream, and then quietly hand off almost all of it back, adjusted down to the last ion. In this module, we're going to unpack how they do that, and why it matters far beyond just making urine. Because the kidney isn't really a waste disposal organ. It's a master regulator of your entire internal fluid environment, its volume, its salt content, and its pH. Let's get into it. Here's the map for the next 20 minutes. We'll start with how the nephron is built and how it makes urine. Then how it concentrates that urine using a clever trick called the countercurrent multiplier. From there, we zoom out to the body's fluid compartments, how water and sodium are balanced, and finally how the kidney and lungs team up to defend your blood pH. By the end, you should be able to classify any acid-base disorder and predict how the body fights back. 6 objectives, don't memorize them now, just know this is where we're headed. So before any mechanism, the big picture, the why. The kidney solves 4 homeostatic problems simultaneously. 1. It clears metabolic waste, urea, creatinine, uric acid, and a lot of the drugs you take. 2. It sets your blood volume and blood pressure by deciding how much water and sodium to keep versus dump. 3. It holds each electrolyte, sodium, potassium, calcium chloride, phosphate, right at its set point. And 4. It defends blood pH near 7.4. Here's the key insight for the whole module. The kidney accomplishes all 4 of these using the same basic machinery, forming urine. Change what ends up in the urine and you change the blood. Everything from here is just the details of how it decides. The functional unit of the kidney is the nephron, and you have about 1 million of them per kidney. Let's trace the path. Blood arrives at a little tuft of capillaries called the glomerulus, sitting inside a cup called Bowman's capsule. Together, that's the renal corpuscle, and that's the filter. Fluid squeezed out of the blood here becomes filtrate. That filtrate then flows through the proximal convoluted tubule, which does the bulk of the reabsorption, around 65%. Next, it dips down into the loop of Henle, which builds a salt gradient we'll come back to. Then the distal convoluted tubule, where hormones fine-tune sodium, potassium, and calcium. And finally, the collecting duct, where the last of the water is recovered. Notice the theme. Each segment has a job, and its structure fits that job. Structure previews function all the way down. Urine formation is 3 processes stacked in sequence. First, filtration. Pressure forces water and small solutes out of the glomerular blood into the tubule. It's non-selective. It filters by size, not by what's useful. Second, reabsorption. The tubule reclaims the good stuff, glucose, amino acids, and about 99% of the water and ions, and returns it to the blood. Third, secretion. The blood actively dumps selected extras, certain wastes, drugs, potassium and hydrogen ions, into the tubule for disposal. So what you actually pee out equals what was filtered, minus what was reabsorbed, plus what was secreted. Hold on to that equation. It's the logic of the entire kidney in one line. Let's zoom into step 1. Filtration is driven by a tug-of-war of pressures, what physiologists call Starling forces. Pushing fluid out of the capillary is glomerular hydrostatic pressure, about 55 milliliters of mercury, high, because of how the arterioles are arranged. Pushing back in are 2 forces, the fluid pressure already in the capsule, about 15, and the pull of plasma protein staying behind in the blood, about 30. Netted out, 55 minus 15 minus 30 gives a net filtration pressure of only about 10 millimeters of mercury. A small number, but it drives an enormous output. because the filtration membrane is huge and extremely leaky to water. That membrane has 3 layers, and together they let water and small solutes through while keeping blood cells and proteins in the bloodstream. That's why healthy urine has no cells and essentially no protein. Add up all that filtration across both kidneys and you get the glomerular filtration rate, or GFR, about 125 milliliters per minute. That's roughly 180 liters a day, your whole plasma volume filtered about 60 times over. Obviously, you don't lose 180 liters of urine, so reabsorption has to claw back more than 99%, leaving only about 1.5 liters to leave the body. GFR is the single most important number for kidney function. It's what your doctor estimates from a blood test. And because so much depends on it, the body works hard to keep it remarkably steady even as your blood pressure swings around. Which raises the question, how? 3 layers of control. The first is autoregulation. The kidney managing itself, no outside help. It has 2 tricks. The myogenic response, where an afferent arteriole that gets stretched by high pressure simply constricts to damp the surge, and tubuloglomerular feedback, where a sensor called the macula densa tastes the salt in the fluid leaving the nephron and adjusts the arteriole accordingly. Together, these hold GFR steady across a wide pressure range, roughly 80 to 180. The second layer is neural. In an emergency like blood loss, the sympathetic nervous system clamps the afferent arterioles down, dropping GFR on purpose, to redirect blood to your heart and brain. And the third is hormonal, the renin-angiotensin-aldosterone system, or RAAS. When pressure drops, the kidney releases renin, kicking off a cascade that constricts vessels and tells the body to retain salt and water, restoring volume. Autoregulation handles the minute-to-minute, RAAS handles the sustained problem. Now reabsorption isn't uniform. Each segment specializes. The proximal tubule is the workhorse, about 2/3 of the sodium in water, and essentially all of the glucose and amino acids get reclaimed right here. In the loop of Henle, the descending limb reabsorbs water only, so the filtrate gets saltier. The ascending limb does the opposite. It pumps salt out but blocks water, so the filtrate gets diluted. Then the distal tubule and collecting duct are the regulated zone. This is where hormones make fine adjustments to sodium and water, and where potassium and hydrogen ions get secreted. So the front end does bulk recovery on autopilot, and the back end does the precise hormone-controlling tuning. Keep that division in mind. It's why the same hormones keep showing up at the distal nephron. This is the slide people find trickiest, so let's take it slowly. The problem the kidney is solving to make concentrated urine. It needs a salty environment deep in the kidney to pull water out of. The loop of Henle builds that gradient, and here's the mechanism. The descending limb is permeable to water, but not salt. So, as filtrate goes down into saltier surroundings, water leaves and the filtrate concentrates. The ascending limb is the reverse. It's waterproof, but it actively pumps out salt to the surrounding tissue. Now here's the clever part, because the two limbs run right next to each other in opposite directions. The salt pumped out by the ascending limb makes the fluid entering the descending limb even saltier, which loads more salt to pump out next, and the effect compounds down the loop. That's the multiplier. The result is a standing gradient from about 300 at the top to 1200 milliosmoles at the very bottom. The kidney has basically built a battery of saltiness, and next we'll spend it. That gradient is just potential. A hormone decides whether to cash it in. That hormone is ADH, antidiuretic hormone. When you're dehydrated and your blood gets concentrated, the pituitary releases ADH. ADH inserts water channels, aquaporins, into the collecting duct wall. So as filtrate passes down through that salty deep tissue, water is pulled out and returned to the blood, and you produce a small volume of concentrated, dark urine up toward that 1200 number. Flip it around. When you've just chugged a bottle of water, 88 switches off. The collecting duct stays waterproof, and water flows straight through a large volume of pale, diluted urine, as low as 50 millimosmoles. One hormone acting like a dial, spanning a 24-fold range of urine concentrate. That's the payoff of the loop machinery. How do we actually measure any of this in a living person? The tool is renal clearance, the volume of plasma completely cleared of a substance each minute. Clearance equals the substance's concentration in urine, times urine flow rate, divided by its concentration in plasma. The trick is picking the right substance. Inulin is the gold standard. It's freely filtered and neither reabsorbed nor secreted, so its clearance equals GFR exactly. In the clinic, we usually use creatine instead. It's already in your body and close enough. And by comparing any substance clearance to inulin's, you can deduce what the tubule did to it. Glucose has a clearance near zero because it's completely reabsorbed, while something secreted will clear faster than it's filtered. Clearance turns the invisible into a number. Let's pull back from the kidney to the fluid it's regulating. About 60% of your body mass is water, roughly 42 liters in a 70-kilo adult. 2/3 of that sits inside your cells, the intracellular fluid, about 28 liters. The remaining third is extracellular, about 14 liters, and it splits again. Most of it is interstitial fluid bathing the cells, around 11 liters. And a smaller piece is the blood plasma, about 3 liters. Here's what matters. The kidney directly adjusts only the extracellular fluid. But because water moves freely across cell membranes, changes there ripple inward to the cells. So the kidney manages the ECF, and the ICF flow follows. Lock in those fractions, 2/3 in, 1/3 out, because they explain everything on this next slide. So what decides how water splits between these compartments? Osmosis. Water follows solute. And since sodium is by far the dominant solute in the extracellular fluid, sodium effectively governs where the water goes. Cell membranes let water through freely, so the two compartments always equalize at about 300 milliosmoles. Change the extracellular tonicity and water shifts. If the ECF becomes hypotonic, too watery, water rushes into the cells and they swell, maybe even burst. If the ECF becomes hypertonic, too salty, water is pulled out and the cells shrivel. This is the crucial takeaway, because sodium drags water with it. Controlling sodium balance is controlling water balance. This is why the next slide is really about both at once. Over a day, water in has to match water out, about 2.5 liters each way. Intake comes from what you drink, water in your food, and a bit of metabolic water from your cells. Output is urine, the adjustable part, plus obligatory losses you can't avoid through skin, breath, and feces. The behavioral half of this is thirst. Osmoreceptors in your hypothalamus are exquisitely sensitive. A rise of just 1 to 2% in plasma osmolarity is enough to trigger them. And they fire two responses at once, consciously. They make you feel thirsty and go get a drink, and hormonally they trigger ADH release so the kidney holds on to water. Intake and conservation, defended together from a single signal. Two hormones tune the extracellular fluid, and they pull in opposite directions. Aldosterone, from the adrenal cortex, is the salt-retaining hormone. It tells the distal nephron to reabsorb sodium, and water follows sodium, while secreting potassium in exchange. So aldosterone raises your blood volume and pressure, and lowers your plasma potassium. Its counterweight is ANP, atrial natriuretic peptide, released by the heart's atria when they're stretched by too much blood volume. ANP does the opposite. It promotes sodium and water excretion and shuts down renin and aldosterone. So volume and pressure fall. And notice potassium is regulated mainly through that aldosterone-driven secretion, which really matters because potassium sets the excitability of your nerves and heart. Too much or too little is genuinely dangerous. Last big theme, pH, held in the tiny window of 7.35 to 7.45. The first line of defense is the bicarbonate buffer system. This reversible chain links CO2 and water on one end to hydrogen and bicarbonate ions on the other. Watch what it does under attack. Add acid, extra hydrogen ions, and reaction shifts left. Bicarbonate soaks up the hydrogen to form CO2 and water, and the pH drop is blunted. Add base, and it shifts right, releasing hydrogen to push pH back down. The genius of this particular buffer is that both ends are under physiological control. The CO2 end is managed by your lungs and bicarbonate end by your kidneys, which sets up the final idea. Two organs move the two ends of the equation on very different clocks. The lungs control CO2, the acid side, and they're fast, acting within minutes. Breathe faster to blow off CO2 and raise pH. Slower to hold CO2 and lower it. The kidneys control bicarbonate and hydrogen excretion. Slow, taking hours to days, but far more powerful and complete. Here's the rule that ties it together. Whichever system caused the problem, the other one compensates. A lung problem gets a kidney fix. A metabolic problem gets a breathing fix. Fast responder and definitive responder, working as a pair. Now we can classify any acid-base disorder with just two questions. Question 1, is the pH too low, acidosis, or too high, alkalosis? Question 2, is the root cause respiration, meaning CO2, or metabolic, meaning bicarbonate? 2 by 2 gives 4 boxes. Respiratory acidosis, you're underbreathing and retaining CO2. The kidneys compensate by hanging on to bicarbonate. Respiratory alkalosis, you're overbreathing, blowing off CO2. Metabolic acidosis, you've lost bicarbonate and gained acid. Classic example, diabetic ketoacidosis. And the lungs compensate by hyperventilating to blow off CO2. The metabolic alkalosis, often from vomiting up stomach acid where your lungs slow down. Read the primary problem off the pH and cause, then predict the opposite system's response. That's the whole diagnostic game. Let's land the plane with 4 numbers to walk away with. GFR, about 125 milliliters a minute. Total body water, around 42 liters. 2/3 of it inside your cells. Blood pH 7.35 to 7.45. And the medullary gradient the loop can build up to 1200 milliosmoles. But the one idea to keep above all the numbers is this. The kidney forms urine in order to regulate your entire internal environment. Its volume, its electrolytes, and its pH. Filtration, reabsorption, and secretion aren't the point. They're just the tools it uses to defend all 3. That's Module 7. If this helps, leave a comment and like what you see broken down in the box next. I'll see you on the next one. Have a good day.